Welcome to our poster presentation titled idgreactome.org, a web-based platform for visualizing dark proteins in the context of reactome pathways. My name is Tim. I'm a member of the Reactome team, and I'm going to show you around. Reactome is a web-based platform that allows the visualization of pathways, complexes, proteins, reactions, and more. And the idg.reactome.org portal provides a comprehensive platform for visualizing dark proteins in the context of their potentially interacting pathways. We define a dark protein as a protein with no known drug targets and very little primary research. The IDG version of the Reactome pathway browser enhances it with new features. It allows the overlay of tissue-specific expression data and pairwise interactions directly onto the pathways in their original diagrams or in a new functional interaction network. We hope that these new features will help researchers generate experimentally testable hypotheses and discover what these dark proteins do. We've also added a new homepage at idg.reactome.org where users can search any protein they'd like. They will be shown a list of annotated pathways in Reactome for that protein, and then will also be shown a list of interacting pathways viewable in the pathway browser. Here you can see the interacting pathways table for the protein CDKL4. By default, we're going to list pathways based on a combined score interactor set. This combined score interactor set takes all of our different features, puts them through a random forest network, and tries to predict which interactors are most likely to be real interactions. You can also click this button here and choose any individual interactor set that you'd like. When you enter the pathway browser from this table, you can click on any pathway you'd like to view. Or you can view the pathway overview. In the pathway overview, you will see pathways highlighted in this pink color. These pathways are significantly hit by interactors of the searched protein, in this case, CDKL4. You can also see that the interactor set is the combined score interactor set. And we're showing all pathways that interact with an FDR of less than 1. You can either click on any of these individual nodes or return to the home page and select an individual pathway to view it inside the pathway diagram. Here inside the pathway diagram, you'll also see highlighted entities. Here's an example of a highlighted complex and a highlighted protein. When you have a highlighted complex, that means that one of the proteins in this complex interacts with the searched protein CDKL4 in this set of interactors. You'll also notice that these complexes and proteins have been recolored from their original color here. By default, we're showing proteins' target development levels. This is meant to give an understanding of how much research has been done about individual proteins. If you look at this complex, you'll see that it is divided into many different striped bands. These striped bands are meant to represent the target development level of individual proteins in the complex. You can also overlay tissue-specific expression data, and it will recolor these entities. We've also added a functional interaction view, which you can see here, which provide the proteins of this pathway as the nodes and interactions as the edges between these proteins. Here you can also see the interacting proteins are highlighted in this pink color, and the node has been recolored to match the current overlay. Again, by default, it's going to show the target development level, but if you were to add a tissue-specific expression data set, these nodes would be recolored. On top of this overlay, you can also overlay interactions directly onto the pathway diagram. Here you can see what that looks like. Inside the pathway diagram, there will be a decorator in the top right-hand corner of complexes and proteins that note the number of interactions available in this data set for this protein, or for this complex, or this protein here. You can click on this decorator and it will open our pairwise pop-up window where you can see the original proteins from the complex you opened as circles represented here, and you can see the interactors represented as triangles. Again, in this pop-up, you'll see that the nodes are recolored to match the target development level. So you can see that this protein and this protein have significant interactions with multiple different T dark or understudied proteins. You can also view drug interactions in this pairwise pop-up by clicking the decorator in the top left-hand corner, if there is one. You can see that this complex has one drug-target interaction available for viewing, and those appear as a purple hexagon here. The table below it allows you to visualize the pairwise relationships in a table view. So you can view the diagram source node, the pairwise interactor, the value of the overlay, in this case, it's the target development level, and whether it's a positive or a negative interaction, and where we're actually getting this interaction from. Because there are so many more interactions than we could reasonably display in this network, if you want to add a specific interaction, you can click this view button and it will be displayed. Similarly, you can remove an interactor from this network by right-clicking on it and clicking remove. I hope that you've enjoyed this presentation on the features of the IDG Enhanced Reactome Pathway Browser and the new homepage. If you have any questions, we'd be more than happy to answer them. Thank you so much.